Okay, we are ready to start. Welcome everyone. Welcome back. Thanks for joining us today for Neurolectives webinar. Actually, I'm connecting to the webinar from the brain stimulation conference from Lisbon. So we have been attending the conference in Lisbon and brain stimulation conference this year. A lot of research, a lot of new highlights, a lot of new things that are coming. So I encourage actually you to attend this event whenever you are, whenever you can. The next one will be actually in Japan in 2 years. So you're welcome to attend the brainstem conference. And today we'll be talking about combining transcranial electrical stimulation with cognitive training in working memory improvement. And I'm very happy to welcome our guest speaker. We are honored to have today with us Dr. Sebastien Scanella. Good to have you here. Hello, Sebastien.
Hello, good to be here too.
So we'll begin, like always, with the presentation, and this will be followed by the Q&A period. So, like always, please feel free to submit your questions at any time using our Q&A tool. And please remember to use chat for any technical or other issue, and use Q&A tool for questions. And we'll try to answer all questions at the end of the session. So please let me quickly introduce our speaker, Dr. Sebastien Scanella. Sebastien is a research scientist in the field of neuroergonomics at the Institute of Aeronautics and Space in Toulouse, France. His work focuses on the study of the interaction between the human brain and complex system with the aim of improving human performance in demanding environments such as aviation, space, and other domains. His research is centered on understanding the neural and physiological mechanisms underlying human decision-making, attention, and cognitive control. And through this work, he aims to contribute to the development of new technologies and methodologies to support human performance in safety-critical environments. So welcome, Sebastien, all yours.
Okay, thank you very much, Alvar, for this introduction. So I have the pleasure to present some of my works that I did at Isao Super-Héros, and it will be divided into two main presentations. The one on a recently published paper, which is a complex task training, and the other one about an ongoing project that we have. Both have the same goal, except it's not in the same environments, but it's to use neuromodulation techniques associated with cognitive training in order to improve performance, whether for healthy participants in complicated and difficult tasks, or for patients in which daily life has become complicated because of some impairments. Before talking about these two studies, I would like just to speak a little bit about the neuroergonomic centers, the lab where I'm working on, for you to have a big picture about the work we are doing here. So this center is in Toulouse, like Raphael said, in the south of France, and is within the Institut Supérieur of Aeronautic and Space in Toulouse, where actually it's a aeronautical engineer school, but we have research departments, and our department is within this brand new building, which is the Neuroergonomic Center that we are lucky to have now, where we can have different rooms and materials to conduct experiments. Speaking of materials, we have different environments. We have flight simulators, like homemade flight simulators, 3-axis motion simulators. We have also drones on ground and flying drones that we can make some experiments with controllers. And we also have different light aircrafts where we can stress pilots in real conditions. We have 8 or 9 little aircrafts. And we recently acquired the Twine engine. It's not that light, it's a little bit larger, which is really nice plane, and it's all equipped with cutting-edge neurotheological tools to do EEG, NIRS, ECG, eye tracking, and with most of them embedded in the plane. So we call it like the experimental flying platform, where we can have this real condition to study. Speaking about sensors, we can do brain imaging because we have some EEG devices, like some stationary EEG for like very fundamental researches, but also portable ones to go in the wild, like we say. And also near-infrared spectroscopy, which is another brain imaging technique. To go in very simple description, it's like a very cheap MRI scanner, and you do like a surfacic imaging of the brain activity, but it has advantage of having a good spatial resolution, which makes it complementary to the EEG that have a good temporal resolution. Along with this brain imaging techniques, we do also eye tracking activity with computer-based system, wearable devices, and also embedded eye tracking system, like in our simulator and in the P68 plane I just talked previously. And we do some cardiac activity analysis also, which is for some features of cardiac activity related to the mental workload that we like to assess during simulated or real flight experiments. Finally, we recently, I think it was 3 or 4 years now, new devices to do some transcranial electrical stimulation on pilots, of course, because these pilots are our brain model for us. We are in the aeronautic centers in Toulouse, and we use the pilots to understand how the brain is coping with difficult situation, how the stress is affecting performance, and so on. But not only with pilots, my work is extended to other people in difficult condition, and among them, I work for several years now with TBI, traumatic brain injured patients. That represents some kind of chronic model of impairment, executive impairment. And for both pilots and patient, the purpose of neuromodulation is to increase some kind of behavioral cognitive benefits we may have with different protocols. And this is what I'm going to talk about today. So to start with, I will present the first study we recently published about how we can use neuromodulation, transcranial electrical stimulation, to improve the performance, the behavioral performance in complex task handling. Before going to details, I will just say a few words about the techniques, the neurostimulation itself. It's not quite intuitive, but I won't go into deep details. First, because I'm not an expert on it, and the second, because I think it's more interesting talking about results. But there is a lot of different electrical current stimulation that we can use to facilitate the neural processing. And the mostly widely used, and also historically, it's the TDS, for transcranial direct current stimulation, where using different electrodes, you can stimulate the brain with a direct current, which has a polarity, and depending on the electrode, it can be positive or negative. We used to say that the anodal electrode will provide the positive activity and the facilitating effect, whereas a cathodal one will induce some kind of inhibitory effect over the cortex. And these techniques is meant to change the action potential threshold, the resting membrane potential threshold, and to make the neuron be able to discharge more easy with this kind of stimulation. On the other side, you have the TACS, for the transcranial alternating current stimulation. As you can see, it's some kind of sinusoidal curves where the current is changing from positive to negative in between two electrodes. Let's say you have an anode, what we call an anode here, and then a cathode there. So the current is changing. So as you may understand here, we can no more speak about cathode and anode here, because every cycle the polarity changes. So the cathode is a cathode during few milliseconds, and then it's changing. And this kind of activity, electrical activity, will have an influence on brain oscillation, meaning it will potentialize some kind of oscillation, brain oscillation in the brain. And finally, the one that I am speaking about today is transcranial random noise stimulation. And this kind of stimulation is part of the alternating current stimulation, except that instead of having some kind of constant frequency for the oscillation, let's say 100 Hertz, for instance, here we are swiping, we are using a band pass between, let's say, 100 Hertz and 600 Hertz. And then this frequency is alternating, is changing randomly with a Gaussian distribution to try to have the representation of all frequency. And this kind of stimulation is supposed to induce some kind of neuronal stochastic resonance. So it's still in debate. It's not quite a consensus today, but it's the most probable explanation. That's by inducing noise into the neural network, you will enable to make some subthreshold neurons to reach a supra-threshold activity. Then using this noise, you will kind of make this network in resonance, and then you will you will facilitate some brain activity. So once again, I won't go into detail, just for you to know that this is the technique we used, because it has been shown that this kind of stimulation may improve executive functions. And actually, executive function is it's at the heart of most of my work, because whether it's a complex task you are handling, you need to have some good working memory, planification, inhibition, and patients that I'm working with have this kind of executive function impairments that I like to cope with. We use this stimulation not in any parts of the brain, and we have to choose one brain or two brain areas. And among all the possible targets we were looking at, the dorsolateral prefrontal cortex seemed to be the most promising, because it's a brain area that is a key region for most of executive function. It has been shown to be involved in working memory, inhibition, and it's within what we call the frontoparietal network for attentional control. And so it was a good target for us. And as we wanted to go further a little bit than the literature at this time, we not only targeted this brain area, but we also compared different montages. So just to go quickly about this, conventional montages, what we call SD for standard or simple definition, whether it's TRNS or TSCS or TDCS, the montage could be the same in this standard definition. You have two sponges or electrodes positioned on the brain the brain region you need to stimulate, and most of the time you put the anode electrodes just over the region you want to stimulate, and you put the cathode on another parts. But recently it has been shown that this kind of simple definition montage has induced a widely spread activity, as you can see on this influence map. And you can see that in this conventional montage, this electrical field that we are visualizing here is spreading on this white matter across both prefrontal cortices. So it's kind of blurry stimulation. It's not as focal as we wanted when we are speaking about stimulating the dorsolateral prefrontal cortex. On the other side, this high definition, relatively high definition montage can induce more focalized stimulation. And we use what we call the 4x1 montage, where the F4 electrodes is the stimulating electrode actually, where we have 100% of the current we are using in the montage, and the 4 peripheral electrodes here just receive a quarter, roughly a quarter of the amount of electrode, and this montage makes the stimulation more focal. And here you can see that we are very targeting the right prefrontal dorsal cortices. So in addition to assessing the improvement effects of neuromodulation, we also compare these two kinds of montages, simple definition and high definition. I talked since the beginning, I'm talking about complex task, and what we are referring as a complex task in the superhero is always the pilot in his cockpit with this huge amount of displays, nodes, and he has to do a lot of complicated things. But before going into this plane, we wanted it to assess it with some kind of simplified complex task, which is supposed to solicitate the executive functions. And actually, we found this Space Fortress game, which has been developed a long time ago, and it was meant to train pilots, actually, at the beginning, to do some kind of multitasking. At this time, a researcher understood that flying activity requires multitasking executive functions, and they understood that training it may improve the flying management for the pilots. And in this task, you are controlling your ship here in blue, and you have to kill the Space Fortress, which wants to kill you in return, of course. So you have to avoid missiles and throw missiles to the fortress, but at the same time, you have to do some subtasks. And among them, you have some kind of bonuses. What we call bonuses, it's a memory task, like end-back task, if you have already do this kind of task, where you have to recall specific characters and push a button whenever they appear. You have some identification of minds. You have to say if they are for type 1, type 2, and so on. And you have other different subtasks. Just keep in mind that we have some kind of working memory, inhibition, and switching, alternating this subtask and doing all in the same time in a multitasking environment. So it is really supposed to involve executive functions. So the question was, can we improve Space Fortress score? Can you improve Space Fortress performance with brain stimulation? To test this, we train participants during 3 days. So they came at the beginning to have some kind of baseline measurements. So we did Space Fortress during 10 minutes just to have the baseline score, how good they are at the beginning without any stimulation. It was for the game session 1 here, the GS1. Then during 3 days, they had this game Space Fortress training with brain stimulation or not. It was for the 3 groups, the sham group with a placebo stimulation, simple definition, and high definition TRNS groups. And during 6 game session, GS2 to GS7, they received concomitant brain stimulation. It was two times 10 minutes. So it was 20 minutes practicing the game and having the stimulation at the same time, a real one or a sham one. After these 3 days of training, they came back the 5th day to have some short-term evaluation. So they practiced the game again, but without any stimulation. and then 10 days later to try to analyze, to observe some kind of long terms, let's say middle terms effects. And thanks to this design, we can evolve short term between baseline and short term, long term between baseline and long term, and some kind of what we call retention effect, meaning after the stimulation has been stopped, is there a difference between the day just after the stimulation and 10 days later? This stimulation has maybe induced some kind of long-lasting effect that we may observe later on, and that's what we were interested in. To summarize this hypothesis, so we wanted to observe a better improvements in space fortress scores with HGTRNS compared to other groups. But these improvements in space fortress scores could be observed in different manners, and the sub-hypothesis could be an effect of this stimulation on what we call the learning effect, the learning rate, meaning is this group learning faster than the other group? The behavioral performance is increasing faster than the other, so it will be all along the training session. Or it could be just a short-term effect, meaning that stimulation have an effect just after the training, but it would disappear few hours or day later, maybe. But it could be also long-term effect. Maybe it's not observable right now, but this cerebral plasticity or whatever mechanisms have been solicited need some time to take place, and we will observe them a little bit later. And finally, another alternative hypothesis could be that there is some consolidation effect, and it will be just between these two sessions. For the rest of the statistical analysis I will present, we also take into account some parameters and some variables, and like the game experience, because we know when we are dealing about gaming that people are not equal in the population. Some are really pro gamers, like we say, they spend several hours a week, and some have never played any game. So we had to take this experience into account in analysis of covariances. Let's go to the behavioral effects of this stimulation. So I'm presenting here the total score. So this is the global score at Space Fortress according to the game session. And here on the upper part of the panel, you have D1 to D15. In gray, this is the score for the sham group with no stimulation. In blue, same for the simple definition TRNS group. And in red, we have the HD TRNS group. So the first alternative hypothesis was about the learning effect, how fast these groups are learning. And you can see that we have this higher trend for the HD TRNS group with a coefficient here which seems to be higher than the two other. Unfortunately, when we are looking at this into details, it didn't reach significance. We were at 0.9, probably due to a small sample size in each group, meaning that even if the HGTRNS group is a little bit higher in terms of learning rate, sham group has such a huge distribution with some people very, very high in the learning rate that made this effect non-significant. However, the game experience was significant, meaning the higher the game experience at the beginning of the training, the higher the learning rate, the faster the learning rate, if you want. Second hypothesis was about some short-term effect, meaning can we have an effect of this stimulation just right after the training? And the answer for us was no, because we see we didn't have a significant group effect, meaning the difference in this group is not significant, with still some kind of gaming experience effect. The third alternative hypothesis was about the long term, and here things are became to change a little bit, because what we found is that the gaming experience did not influence this long-term performance. Remember, it's a delta. It's a comparison of the 11th game session compared to the first one. And here we found that the group effect was significant, but post-hoc analysis showed us that it was only because the HDT-RNS group was higher than the SDT-RNS group, but not compared to the sham group. And we could talk a little bit later on about on this particular effect, but I will first present the consolidation effect. So finally, what we found is that when we compared the long-term and short-term performance, the difference between these two sessions, gaming experience did not influence at all, but we found some group effect with the superiority of this high-definition group compared to the two others. Meaning, if we go back to this main graph, that's looking at the last performance of the short term compared to the last one of the long term, HDT-RNS groups tend to keep on improving themselves. They benefit from some residual effects of the stimulation, whereas sham group and SD group did not. Okay, so that was for the healthy participants and in terms of complex task training. And what we wanted to know was whether we could use the same stimulation parameters to help some patients, traumatic brain injured patient, to improve behavior in the daily life. And we worked with the hospital of Toulouse with specific some patients and try to assess the interest of using neuromodulation for them. And this is what we did. So just some words about TBI patients. These are patients that have some traumatic injuries in their brain, and because of the blood in the brain, there were some degradation, some lesion in the brain, and we selected the patients, the TBI patient that has what we call dysexecutive syndromes. Quickly, dysexecutive syndromes, it's all about executive function impairments, meaning they have difficulties with working memories, with inhibition, with some perseverative behavior. Planning activities is very difficult. And at the end, these patients have some difficulties to behave normally in daily life. Most of the time, it's very difficult to have a job because of these impairments. There is some rehabilitation for these patients. Of course, they are involved in some protocols to try to recover as much as possible from their syndromes, but they seem to be limited, and also there is some difficulties to assess the recovering in real life. They are trained with some laboratory tests, some executive function tests, but either they are progressing, they have some improvements in this task with no transfer to the real life, or they found some strategy to cope with this task and have some significant improvements that are not really here. So we worked with an enterprise in Toulouse, which is called Covirtua, and they are proposing to train this patient not with this artificial task, but with some ecological tasks. And these are some examples of the software that we can use. They are proposing the patients to do some shopping in virtual environments. So it could be with a virtual headset or on a computer. It depends on the compliance of the patients. Or for instance, using a GPS to navigate into streets like in real life. And the interest here is that the patients and the physician are working together to adapt difficulty for the patients and to change events, for instance, increasing some working memory assessment within the task or inhibition and so on. I will just show you a quick video. Unfortunately, it may be a little bit laggy in Zoom, so you can find it with the Covirtua name in YouTube, but just I will comment it here for you to explain how it works. Switching the sound off. So as I said, there is two parts in the software, the patient one and the physician one. And during the training, the patient is facing this virtual environment. For instance, it could be here doing some shopping, and he may ask for help. He may look at different products according to a list, like in real life, and he has to fulfill a list for, let's say, for instance, to prepare some dinner. And the physician may change the environment, may change the difficulty. He may also add some, this is a note, for instance, to assess the flexibility of the patient. Oh, I'm sorry, I forgot to say that there is no butter. Can you add one? So the patient may update the list and may train to do this in this environment. And then the physician can assess it. And this is it. He can ask for help, go into the market. So let's just remember that it's supposed to be real life simulation. It's supposed to be a training of this executive function, but embedded in an ecological environment. And what we wanted to assess is that if this kind of cognitive training can be efficient with some kind of stimulation. And with no surprise, we use the parameters that worked for us in healthy participants with this Ph.D. TRNS montage with exactly the same parameters. One issue, though, with this kind of assessment, so it was about on 14 participants, 14 patients here, is that in conventional assessment in clinical trials, having this group analysis may often lead to misleading or weak effect size in the results, because they have different lesion types, they have different lesions. So it's very an heterogeneous population. So we knew at this time already that group analysis is not suited to evaluate this kind of intervention. So we decided to apply a new experimental design, which is called single case experimental design, or SCED, which has been developed recently, and we are working with cutting edge people in this domain to assess, to use this design. Let's say that this design is compensating the small sample size, because you are working with one patient at a time, by increasing the number of measurement of your variable of interest, but increasing it a lot to have some kind of statistical analysis of the effect of your intervention. Maybe this figure will explain it a little bit better. So there is different types of SCED, and we use what we call the multiple baseline SCED, where you start with the conventional intervention, what we call the standard ergotherapy, meaning that the patient is at the hospital, but he or she is doing regular activity in the ergotherapy. And then among your patients, you will start randomly the moment of your intervention, which is our Covirtua plus Ph.D. TRNS. And you will just choose randomly the patient that is starting during 4 weeks. And then after 4 weeks, whenever, regardless of the moment you started it, after 4 weeks, you stop the intervention, and then you go back to the standard ergotherapy, and you still observe your variable. And your variable, you see, I said that we have to measure it very often. And this is this little downsided arrows here. It has been evaluated twice a week. for a total of 24 times during these 12 weeks. So you understand here that this variable has to be specific because it has to be robust to some kind of repetition effect with a plateau or habituation effects. So we had to choose specific variable that is suited for this CAD. So there is different ones, but we choose one that is called the goal attainment scaling, or GAS. Actually, it's the same people that have developed the SCAD, and they thought about this GAS to create some behavioral measurements that is compatible with the SCAD. So the goal attainment scaling, it's a scaling of behavioral improvements. You are setting some behavioral objective goals with the patients according to the ergotherapeutes, the clinician, and so. And we quote this goal. Let's say we speak about concentration, which is one of the metacognitive executive function. And at the beginning, the patient has impairments of concentrations, meaning she or he cannot concentrate more than 2 minutes. Then you have this scale from -2 to +2 to reach the most favorable results, which is having more than 8 minutes for these specific patients. Don't forget that it's all individual goal. And in between, we have different level. The 0 level, it's not like you could be intuitively, it's not 0 improvement, but it's expected level. So if you stay at the initial level, you stay in -2, then you will progress according to this scale, okay? If we still look at this concentration goal for this specific patient, ideally, it will be something like this. So we start during 3 weeks, so six measurements of this concentration during the standard ergotherapy, okay? Then we start the co-virtua plus stimulation here, and we see that there is improvement in this score for this concentration. And what we expect for this patient is not that the effect disappear as soon as we stop the cognitive training plus stimulation, but that plasticity may lead to some kind of maintaining, and this patient go back into real life with some robust improvements. What we found among the 14 participants, I forgot to say that what is convenient with the SCAD is that we can do statistical analysis, as I said in the beginning of the presentation. And there is different tools, but if you look at one, you may use this TOU sometime by Tarloff. Quickly, it's a kind of non-parametric correlation coefficient, and that will attest with a statistical and an effect size, a statistical p-value and an effect size will attest about the effect of your intervention. Is this evolution of the GAS score influenced by your intervention or not? Among the 14 patients, we found that 10 of them reach at least one goal among the 3 that has been defined for each of them, or even the sum of the 3. And among this behavioral goal that were behavioral activities that they are supposed to do, like concentration, like handling your mailing box, resuming painting, for instance, they were not involving in every kind of executive function, but we found that they were distributed in focused attention, reasoning, planning, and prospective memory. So these results has to be taken with precaution, because it depends of the patient and the objective, the goals that has been fixed. If we had 100 patients with a lot of different goals, maybe this will change a little bit. But for this population, actually, it was this distribution. as a conclusion, we can say that this kind of focalized high density transcranial random noise stimulation may improve the effect of cognitive training, either in healthy population with this complex task as space for rest, with this complex task training. So there is improvement with this kind of stimulation compared to the two others, but it was not necessarily in terms of how fast you are learning, or in a short or very middle term effects of this, but more in terms of consolidation effects. It's like if we had trigger some plasticity in the brain, and then the time this plasticity needs to take place will induce some kind of later effects, but consolidating effects, and maybe longer than the immediate one. And either in this traumatic brain injury patients with a complex task, not really complex for us, but when you have executive impairments, executive function impairments, real life situation may be really tricky. And we found that using this kind of cognitive training, Covirtua plus this stimulation may improve daily life activity for them. This work has been done with my colleague from the Hospital of Toulouse, the Euromove team in Montpellier, and my colleague from the Neuroergonomics Centers, which I thanks for the help. And thank you for your attention, and I'm pleased to answer any question you may have.
Thank you very much. Thank you very much, Sebastien. Excellent presentation, and actually to have your point of view coming from completely different field that we are used to. It's really, really very, very enriching. We are opening room for questions. So we are always thinking about applicability of the results, and piloting an airplane is obviously very demanding task requiring skillful execution of learn procedures, and there is something similar in going to the translational science. So in older adults with simple ability to stand or walk while performing cognitive tasks, example with dual tasking, requires coordinated activation of different networks. And so my question is if you see any applicability of your results of this first part to improve resilience, or the ability of elders, for example, to walk and stand under challenging conditions with your results from the first study in pilots.
So I'm not an expert in elderly people and how cognitive function may decline according to the time, but I've seen some work about it. But what we know, what I know, is that it's also some executive impairments for us. I'm not expert also in motor or other part of the brain impairments, but what I think is that even when we are old, there is still some room for plasticity, even if it's few maybe. But I think that brain stimulation coupled with cognitive training, always, not only alone, but may potentialize a little bit all the thing we are looking, the different tasks we are looking, like doing mathematics, doing crosswords, doing other things that may stimulate your brain, and concomitant electrical stimulation may potentialize it and may reduce some impairments due to the that have been counted by the age.
Okay, thank you. We have some questions from our audience. So the first one regarding specific parameters you were using in the first parts of the study regarding TRNS. One question from Vitold regarding if you are using the full frequency range in TRNS from 0 to 500, or you were filtering and using specific frequency. And then if you can comment regarding the intensity. And regarding specific procedure, if you are doing like 10-minute stimulation and then a break, and then another 10 stimulation, another break, if you comment on this.
Yeah. Okay, so we in the literature we saw that it has been shown that the best frequency range in TRNS and also in TSCS, but it's a little bit different. But in TRNS, it's between 100 Hertz and 600 or 700 roughly. So we wanted to use it, but we were a little bit limited to 500 Hertz with the device we had at this time. So we use a band filter between 100 Hertz and 500 Hertz, which is the most commonly used in literature. So we applied it. This is for the first part of the question. And then the intensity was exactly as you see on this figure, between minus 0.5 milliamperes and plus 0.5 milliamperes. And you see with a little fade in and fade out to help the scalp for habituation effect, the tingling effect, the little tingling effect we had. I think maybe I'll forget the last part of the question, the third part.
If there was the break during.
So what we did, maybe here it's more obvious. So there were no break. We split in 2 sessions, but it was artificial because actually it was just for other behavioral analysis, but it was 20-minute stimulation with 20 minutes, 2 times 10 minutes. But when the first 10 minutes was finished, then the participants played immediately the second session of Space Fortress. So it was 20 minutes plus stimulation each day.
Okay, and there is another regarding the parameters. How do you compare to sham in the TBI study? Actually, no, this was regarding TBI from Irving.
Mm-hmm. Yeah, actually the sham was exactly like this fade in the TRNS, but it immediately fade out like the end here. So it was during, I'm not sure, it was 20 or 30 seconds of increasing and decreasing stimulation, but with exactly the same parameters than the stimulated group.
Okay, there is another question from Valerie. What would change if you use TMS instead of TS? Obviously, yeah.
A lot of things. Yeah. My last experience with TMS was 12 years ago. So I may not be the best person to insert that. But actually, I recently read a paper, I think it was from Hantal and Herman in 2016, something like this, that using some parameters in TACS, sorry, you may approach with pulse mode TMS, but that just for the record. Right now, I think that using some kind of high frequency repeated TMS, in high frequency more than 5 pulse per seconds, because TMS high frequency tend to increase excitability, and low frequency close to 1 Hertz pulse in TMS tend to decrease. So short answer would be with some parameters of TMS over the right prefrontal dorsolateral cortex, we may find comparable results, but with a lot of difference because TMS is supra-threshold and it's creating stimulation. But I think that we have this TMS effect that will increase network efficiency with the DLPFC that we reach some kind of with the TRNS, the AG montage.
Great, really great answer. Question from Jesse. If you were using or you plan to use actually some imaging techniques like fMRI, EEG, or NIRS, then there was a question before and after stimulation or before after the training to have somehow information from the neuroimaging point of view about what's going on.
To be sure, meaning that can we observe the immediate or long-lasting effect at the functional level or the anatomical level in the brain? I'm not sure that these effects are large enough to be observed at the anatomical level. I'm pretty sure that it's not that huge. That could be on the functional level. However, maybe we can observe, but I will more bet on like EEG techniques to observe this, maybe on the frequency domain, some kind of alpha or theta bandwidth, for instance, or even we are currently working on the rested state EEG parameters like microstates parameters to see if something has changed at this functional level in the brain and at rest. So with fMRI, I think it's not sensitive enough, but with EEG, maybe it could be interesting to assess it and to visualize it.
Okay, there is another question from Alan. What type of cognitive remediation task are you using?
What kind of cognitive remediation task?
Yes.
Meaning that about co-virtua or about a classical one?
This was regarding virtual, I guess, because it's the timing that we are talking about the virtual task with TBI.
Yeah. Actually, these are the tasks we are using. It's doing the shopping and adapting the shopping list according to the physician that is trying to help the patient to do the maximum, the best he or she can do in this shopping environments, and trying to solicitate this executive function, where adapting working memory, switching from one task to another, inhibiting the former product for the newer, and driving also, because we know that we need some kind of spatial representation. We have to conduct a car, which involves working memory and inhibition, for instance. So I'm not sure that was exactly the question that has been asked, but this is co-virtua, and if you go on the website, you will see all details of this. There is a lot of different tasks in the battery.
Okay, the question from Kim regarding the design of the study in terms of blinding, because especially with TRNS, the sham actually it's quite difficult, or it's even more difficult than with TDCS. If you were using the blinding, or you are planning to blinding, how to evaluate whether participants correctly determine which group they were in by post-experiment questionnaires.
Yeah. So our assessment, the things that we did, is assessment of which group are you stimulated or not, but there is no significant correct response for them. So that was a good thing for us. But in terms of protocol, what we did actually is, like you see, we have two kind of montages. So if you just, it was double blind, meaning that both experimenter and the participants didn't know in which group they were. But to be sure that the experimenter understood, did not find, because it was HG compared to the two others, we made him believe that they were also a sham HG group, which was not true. And we split in two, this one with fake sham HD. At the end, the experimenter thought it was 4 groups, 2 sham, 1 sham in SD, 1 sham in HD, and 2 stimulated group, 1 in SD and 1 in HD. And it was all embedded in the NIC software experiment. So you just say that you want a double-blind experiments. You put a password to be sure that nobody is tweaking or cheating, and then we just did a list randomly distributed for the groups, and then we were sure that the both participants and experimenter did not find which group it was.
Perfect, thank you. And another question from Kim regarding the complexity of the game. Actually, Space Fortress is a very complex game demanding attention, working memory, executive and motor control. And Kim is asking if it's possible that the complexity of the game prevented actually you from seeing the benefit of TES, because it affected different participants in different cognitive domains.
That's true. That's true. Ideally, we will have also assess classical laboratory executive function with Stroop, NBAC, go no go, and all these different. But we wanted here to have a neuroergonomic approach where the fundamental aspects are less important. They are always important, but they are less important than the practical approach of having this brain at work, involved in using this executive function in the whole environment of space fortress. So we found significant effect, meaning that involving all this executive function and stimulating the right prefrontal dorsolateral cortex, which is involved in working memory and inhibition that are solicited here, was efficient. And actually, what we found, I didn't present the results here because it was a little bit too much for the time, but we also looked at the score for each subtasks. And what we found actually is that not all subtasks were improved by the stimulation, but it was only the global parameters, the global score that was affected, meaning that the HD-TRNS may have had an effect on the multitasking involving all this executive function rather than on unitary executive function, like it can be assessed with this working memory task, for instance.
Thank you very much. There is actually a repeated question regarding neurofeedback. If you were using or you have experience of using neurofeedback to obtain improvement in executive function or combining neurofeedback with transcranial electrical stimulation.
Mm-hmm. That's very interesting one. And this is a project we have with Frédéric Dehaye from my lab. He is working on neurofeedback and we found some improvement of it. So I will say it maybe in a few months or a year when we manage to do it. But our hypothesis is that neurofeedback plus this stimulation may potentialize the results and improve behavioral results. Yeah.
Okay, and the last question from Alan regarding side effects of using TRNS. I think this is like comparing to TDCS, if there are any differences you can find, or it's at the same level in terms of sensitivity to the stimulation. So on the practical point of view, I would say it's equivalent. I have no knowledge of differences between the two techniques. But on the more quantitative point of view, what I would say is that when you have TDCS, you have this constant 1 milliampere amplitude. And actually, I have to check, but I think that the total amount of current that has been used in TDCS like this for the same montage will be higher than the TRNS one, because here we spend a lot of time around 0 milliampere, and actually the mean of this signal is 0. So I may say that the dosage in terms of quantity, like in coulombs, for instance, is smaller in TRNS than in TDCS. That's really great. We are reaching 1 hour of webinar. So thank you, Sebastien, very much. Thank you for the presentation. Thanks for all questions. Thank you, everybody, for joining us today. I hope you will join us again for the next webinars. So please visit our webpage, neuroelectrics.com, for additional information. And once again, thank you very much. Have a great day. We'll see you next time. Thank you.
Thank you. Thank you for the invitation.
Thank you.
Have a nice day.
Bye.
Bye, everybody.